right, so by the cell cycle, what we mean is the life cycle of a cell. And most of the life cycle of a cell is spent in a phase called interphase. The translation of that, I believe, technically means like between divisions. Inter as in between, so between cell divisions. But it is all the time when the cell is not dividing. Sometimes they'll show this sort of in a pie chart. This will be mitosis when the cell is dividing, and all of this other part here will be interphase. So basically 90% of the time. It is when the cell is doing all of the jobs that a cell normally does. So making proteins, transporting stuff, whatever the job of that cell is. But interphase itself is then subdivided into 3 subphases. And they are G1, S, and G2. And so sometimes you'll see that same pie chart like this, and you'll see G1, S, G2 as the subdivisions of interphase. Again, it's still interphase, all of this, all the way around. But it's sort of subdivided into those 3. So let's talk first about G1. So G1 stands for primary growth. This is when the cell has first been born. Think of a brand new cell. It's probably going to be smaller because the cell just came from a cell that was dividing. It needs to probably make some more mitochondria, build some more membrane, make some more cytoplasm. So the cell is going to grow mainly during this particular phase. It's also going to be doing whatever the jobs of that cell are going to be. If that's a skin cell, it's going to do what skin cells do. If it's a bone cell, it's going to do what bone cells do. If the cell is never ever going to divide, it will never go past G1. In fact, you'll learn this tomorrow, but it'll usually, once it's done growing, go into a phase called G0, which is sort of like this phase where the cell will never divide again. Your brain cells, for example, would be in G0. Some cells can go back and forth. Liver cells, from what I read, are usually in G0, but if you had a traumatic injury or you donated a piece of your liver, because you can actually donate a piece of your liver, just like you can donate a kidney, your liver could come out of G0 and those cells could go back into reproducing again. Olivia, did you have a question? No. All right, if the cell is going to divide at some point, then the next phase it will go into is called S. S is where the chromosomes replicate, meaning they make a copy of themselves. So they're literally going to go here from being sticks to being X's. This is the part we talked about last week, how a chromosome originally looks like this. But if the cell is going to divide, it makes an extra copy. What are the 2 sides called? Do you remember? Yes, chromatids. Good. Sister chromatids sometimes. Those are the 2 matching sides. So that's only going to happen if the cell plans to divide. If it's never ever going to divide, like I said, this will just go into a stage called G0. The chromosomes will always be sticks. Synthesis, the chromosomes make a copy of themselves. G2, the cell now starts to make the enzymes and proteins that will be necessary for it to divide. And again, all 3 of these are subdivisions of interphase. So all of this is considered interphase. The cell is not actively dividing, and this is 90% of the life of the cell. Originally, and you don't have to know this for a test, but G1 and G2 originally stood, instead of being thinking G for growth, which makes sense, it actually stood for gap 1 and gap 2. Because back in the day, the microscopes they had, if you use the microscopes we have now, when the cell is dividing, you can see the chromosomes, even under a light microscope. They are really obvious, big, thick-looking X-shaped things. It's obvious something's going on. It looked like when the cell wasn't dividing, since the chromosomes, remember, the DNA when a cell's not dividing just looks kind of like gray. It would just be like a grayish area here, like this. It just looks kind of like this. Basically, they just assumed nothing was going on. Since with the microscopes they had, they couldn't see anything, but when the cell was dividing, they could see stuff. They just called it gap 1. There was this of time where nothing was happening. We now know during that gap of time, that's when the cell's doing all of its jobs, protein synthesis and all that, but the microscopes they had were not good enough to see what was going on. So they just assumed it was a gap. A cell that steps on another cell's foot? Mitosis. Thanks, Ashley. Everyone, I am recording. You realize everyone is now going to hear your very bad joke.
So wait, it's 90%. It's like 90% of the time.
No, 90% is an interphase as a whole. All right, and mitosis, that's when the nucleus divides. And then cytokinesis is when the rest of the cell divides. So mitosis, technically, a lot of times they will simplify and they will say that mitosis is asexual reproduction, asexual cell division. If you want to get really, really technical, mitosis is not division of the whole cell. It only refers to the nucleus dividing. However, in almost every cell, at the end of mitosis, while the last phase is going on, the rest of the cell divides too. Are there cells where mitosis happens without cytokinesis? Yes. A really good example of that are muscle cells. Muscle cells undergo mitosis, but not cytokinesis. So if you take an anatomy, you may know this. So if you look at skeletal muscle cells under a microscope, they're really long and skinny, and you'll see all these little dots. Those are all nuclei. In other words, they undergo mitosis and make more nuclei, but the cell itself never divides. So it's not a disorder. There's not an extra set of chromosomes in one nucleus. Each nucleus has the correct number, but there's multiple nuclei. Why would it want to do that? Well, if you think about it, muscles are mostly protein, and the DNA is the code for making proteins. So if you're going to have a big, long cell, it kind of makes sense to have lots of nuclei so that you can have lots of directions going out to continue making protein versus one nucleus that's trying to control all of this long cell. So anyway, most of the time, mitosis and cytokinesis go together, but in theory, and it's always a test question, I guarantee you it'll be a test question. If mitosis happens but not cytokinesis, what do you get? You get a cell that has two nuclei. The nucleus has divided, so you end up with two nucleuses, but you don't have two actual full complete cells. All right. So we're going to run through the stages of mitosis. That's the rest of this. This is just, again, a picture. Again, see, notice how most of the life of the cell is spent in interphase and just this little segment where it divides. Yes?
We're supposed to put in that little box in the outline.
I don't know.
It just says, like, insert diagram here showing before S and after S.
Oh, before S and after S. Actually, no. Then what you want to put there is that before S, the chromosome looks like this. And then after S, the chromosome looks like that. That's what you want in that box. That's where the chromosome makes the extra chromatin. It's not a separate phase. It happens during the end of mitosis. So sometimes it's shown as a separate thing, but technically it's happening during the last phase of mitosis at the same time. All right. So let's go through the stages of mitosis. In theory, mitosis can't necessarily be divided into 4 stages. We simplify it by saying PMAT, Prophase, Metaphase, Anaphase, Telophase. You may remember learning it in bio, PMAT. But in theory, it's possible that if I gave you a microscope and I said, look at these cells, you might find a cell that was actually in what we would call pro-metaphase. It's not quite to metaphase yet, but it's not quite the textbook picture of prophase either. They would never ask you on the AP exam to identify a cell that was in between phases. But just keep that in mind. It's actually a continuous process, but we're going to sort of subdivide it into 4 distinct phases. So the first phase is prophase, which you're going to see in prophase, and I'll draw a little sketch of it. I'm going to draw you a cell that has 4 chromosomes, because if I was going to do a human cell, I'd have to draw 46 chromosomes, and that would take way too long. So if you think of it, this is what prophase would sort of look like in a cell. Here's your cell. This is the nucleus. The nuclear membrane sort of disintegrates, and the nucleolus disappears too, so you don't see it anymore. When they say the chromosomes condense, it means they coil up, because remember, normally you can't actually see distinct chromosomes. The DNA just looks kind of like a tangled mass. So I'm going to draw the chromosomes here. So here's, I'm going to draw, like I said, I'm going to have a cell that has 4 chromosomes. And then when they talk about the centrosome, animal cells have these things called centrioles. They look kind of like this. Plant cells don't have those, but they still have the area where those would be located called the centrosome. What's going to happen eventually is these are going to migrate to opposite sides. So in the next phase, you're going to see the centrosomes and the centrioles are going to be, this is what the next phase is going to look like. So they're eventually going to end up on opposite sides of the cell. But right now, they're just sort of starting to move. And the other thing that starts to happen is these little spindle fibers start coming out. So it looks kind of like spider webs here. All right, and that's basically what prophase looks like. And you don't necessarily have to sketch it. The only phase I think that they would want you to know for sure on an AP exam is metaphase, the next one. But it may help to have this idea in your head so that you kind of understand where it's coming from and where it's going. So this is prophase. Nucleus is disintegrating. You can see the chromosomes, physically see them. These spindle fibers are starting to form. And this area called the centrosome is forming. In animal cells, it actually contains an organelle called the centrioles. In plant cells, it just looks kind of like an empty space that the fibers are coming from, but it is a distinct area. Any questions on prophase? It's pretty straightforward, and you should have learned this before. Hopefully it looks vaguely familiar to you. All right, then let's move on. So metaphase is going to be our second one. So this is metaphase. This is the most easy to recognize, and this is definitely the one I would ask you for on a test. So in metaphase, notice what's happened here. These chromosomes line up on what we call the equator. Sometimes it's called the metaphase plate, but if you remember equator, just like the center line of the Earth is the equator, and these two sides are sometimes called poles. And the chromosomes are actually now connected to these spindle fibers, like this. Technically, the spindle fibers are actually connected directly to a protein in the chromosome called the kinetochore. You will not be tested on the kinetochore, but it's like a spiky protein, and the spikes are actually what hook to these spindle fibers. But to me, this is very easy to recognize. Think of meta as middle. M for meta, middle. Chromosomes basically line up in a straight line. So in your cells, if I was going to draw a human cell, I'd have to draw 46 chromosomes, all lined up in a straight line to represent metaphase, which again would look ridiculous, but that is what I would have to draw. All right, any questions on metaphase? Okay. The next thing that's going to happen is now the chromosomes are going to get pulled apart, and that's called anaphase. So this is where the chromatids now become their own chromosomes. So here's our cell. Typically starts to stretch out a bit. I'm still going to draw the little centrioles. This has to be an animal cell. And here's my spindle fibers. And here I'll do my chromosomes in red. Now the chromosomes are sticks again, because they've basically gotten pulled apart, and they're basically moving to opposite sides. And they are now going to each one get its own nucleus, and we're going to end up with 2 new cells. Our original cell had 4 chromosomes. Our new cells still have 4 chromosomes. These new cells that are being made are clones of the original. Because remember, when the chromosome made a copy of its information, it was an exact copy. So technically, when these eventually become 2 cells, each new cell will be completely identical. Your skin cells technically should all be identical to each other, and bone cells and blood cells. They are just making exact copies. There's some other spindle fibers. You really don't have to know this part, but they tend to lengthen. This actually helps to push them apart. There's a few fibers that go all the way across, and they actually get longer, and it forces the 2 cells apart. I have a little animation I'll show you after that sort of clarifies this. All right, last slide of mitosis. So the last thing that's going to happen, telophase, and while telophase is happening, cytokinesis. The chromosomes are going to uncoil back to chromatin again. The new nuclear membranes will form. So now we're going to have, it's basically going to look like, in an animal cell, it's going to look something like this. They're going to pinch apart 2 new nuclei. The chromosomes in each of the cells.